Meiosis, using cell division to make gametes such as male sperm or female eggs. The main idea behind meiosis is to start with one somatic cell, and at the end of the process of meiosis, you end up with 4 gametes. We will put the nucleus in purple for this tutorial, and the chromosomes in red. So in meiosis we start with a diploid 2N somatic cell, and we end with 4 haploid cells that are gametes, either sperm or eggs. The reason gametes are haploid is illustrated best like this. Imagine these 2 gametes, one from a male and the other from a female. If they were to meet in the process known as fertilization, this chromosome would be combining with this chromosome to make a cell that looks very much like the original diploid somatic cell. Meiosis also involves a very important principle called crossover. Crossover creates gametes that are unique, one of a kind. No 2 are alike. The fact that no 2 gametes are alike ensures that any population will have a lot of diversity among its members. So let's redraw that original somatic cell in just a little more detail. We'll have a purple nucleus just like before, but this time one of the chromosomes will be red while the other chromosome will be green. We can pretend that the red chromosome came from the mother and the green one came from the father. Okay, so meiosis will produce the 4 haploid gametes, as you know. I went ahead and drew the results of crossover. Basically, these 2 chromosomes kind of exchange segments with one another, as did these 2 chromosomes. So as you can see, each one of these chromosomes is a unique combination of mom plus dad DNA. And that's why you and your brother don't look exactly alike, even though you had the same parents. Diagram meiosis, we will start with a single somatic cell. And we are going to go through one cell cycle of division. But the main difference in meiosis is that there is a second cycle of division. And at the end of this second cycle, we are going to end up with our 4 haploid gametes. So once again, we're starting out with one diploid somatic cell, and we're going to end up with 4 haploid gametes. We'll pencil in the phases. These will look familiar. We'll have growth 1, then somewhere over here, synthesis, then growth 2, then we'll have prophase, metaphase, anaphase, telophase. Then we're going to have cytokinesis. From there we're going to go right into prophase, metaphase, anaphase, telophase, and a second cytokinesis. Notice that there is no interphase at this point. No interphase for round 2. We'll be exploring prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1, prophase 2, metaphase 2, anaphase 2, Telophase II, and the final cytokinesis. G1 is just like for mitosis, where the cell is growing. It's doing its jobs. It's making proteins. Synthesis phase is when it copies the DNA. And if it's an animal cell, it would copy the centrioles. G2 is when it's prepare for actual division. One more thing to note is at the end of the first cytokinesis, we will end up with 2 cells. And I'm not going to bother drawing the DNA in these nuclei. Prophase 1 is really the only hard part and the only new part. So this might be what our cell would look like at the beginning of prophase 1. The nuclear membrane is disappearing. I'm not going to draw the centrioles or the spindle fibers. The important thing to note is that one of the sister chromatids came from the mother, and the other pair of sister chromatids came from the father. Okay, it's in prophase 1 that crossover takes place. I'm not drawing the whole cell, but in crossover, one of the sister chromatid pairs lines up precisely with the other pair, like that. I'm going to draw it a little bit bigger so you can see the results of crossover. So after crossover, you can see that pieces of the chromosomes from one sister have changed to the other sister, like in this part right here. Also, I made an example down here. The location that shows where things crossed over is called the chiasma. And I've only shown 2 places, there and here. But apparently it can happen like 4 to 8 times on each chromosome. And the main point of crossover is to ensure a mixing of the DNA from both parents. I want to illustrate a point. This cell here from a different species has 3 homologous pairs. During crossover would only occur within this pair, and within this pair, and within this pair. You would not expect to see crossover from this pair over to this pair. So we'll say that does not happen. So let's now take a look at metaphase 1, anaphase 1, and telophase 1. So in metaphase 1, the tetrad lines up on the equator. The tetrad is merely this set of crossover sister chromatid pairs. So in anaphase 1, the tetrad separates, and you can see the red sister pair with the green add-ons is going one way, and the green sister pair with the red add-ons is going another way. They are now referred to as bivalents. So there's a bivalent, and there's the other bivalent. And in telophase, the cell pinches in, as you can see. This is very much like mitosis. The nuclear membrane has reformed. The DNA is starting to stretch out and get long and skinny. Spindle fibers disappear, etc. Cytokinesis 1 is pretty straightforward. We end up with a division of the cell's cytoplasm, and we form 2 new cells. Each cell has a nucleus, which may or may not reform. It all depends. So I will put a question mark there. The nuclear membrane may or may not reform. And there I threw in the crossover sister chromatids, and we do not have gametes yet because these are still diploid cells, 2N. We have not yet achieved our objective, which is why we need round 2 of meiosis. All right, let's talk about meiosis, round 2. Please remember that there is no interphase in meiosis round 2. In meiosis round 2, we're going to go through prophase 2, metaphase 2, anaphase 2, and telophase 2. If the nucleus reformed, it will re-dissolve at this time in prophase 2. But at the end, in telophase 2, the nuclear membrane will reform. Now remember, we ended up with 2 cells in the last slide. So I need to draw what's happening with the other set of cells. This will basically be the exact same thing as the above. Okay, so to begin, one of these cells had the red sister chromatid pair, and the other had the green sister chromatid pair. And I'll add the crossover part so we can hopefully keep it all straight. All the other cool things, of course, are happening, like spindle fiber growth and centriole movement. But we're just trying to illustrate what's happening with the chromosomes. Metaphase 2 is very similar in that regards. But you can see in anaphase 2, the sister chromatid finally pulled apart, and now we have single chromosomes. And the same thing is happening in that other cell. Spindle fibers are pulling the chromosomes towards the poles. And in telophase, the cell pinches in. Nuclear membranes reform, etc. Okay, we're about done. We'll talk about Cytokinesis II. So this is where we left off. This chromosome is different than this chromosome. Different from that one and different from that one. So at the end of cytokinesis, we are going to end up with 4 haploid gametes. And each one will have a nucleus, of course. 2 of the gametes will look like this, mostly red, but with a little bit of green from the dad here and there. And the other 2 will look like this, mostly dad, but with a little bit of mom here and there. So here are haploid gametes, abbreviated by 2N. And each gamete will have a unique combination of mom's and dad's DNA. Now imagine if a cell had 23 pairs of chromosomes like a human. The amount of variation is nearly unlimited. So as a quick summary for meiosis, this diploid parent that has a red chromosome from its mother and a green chromosome from its father produces these 4 gametes that are haploid. And each one is unique. And that is why you look different than your brother or look different than your sister. The end.